I have no disclosures. So I'm the director of the Center for Innovating Neuroimmune Disease at Cornell. Our center is NIH funded. It spans Cornell and Ithaca and currently the Hospital for Special Surgery in Manhattan. Innovating Neuroimmune Disease is our favorite name for what many of you probably know as chronic fatigue syndrome. I'm going to do a lot of introduction of this illness because I think a lot of people here know very little about it. It's now really called myalgic encephalomyelitis / chronic fatigue syndrome, or ME/CFS. We estimated that about 67 million people are affected worldwide. And this is actually about the number of people who now are thought to have long COVID. However, the people who have ME/CFS, many of them have been sick for 40 years because it has occurred as outbreaks and it also occurs as sporadic cases. There's either gradual or sudden onset, but unfortunately there's no FDA-approved drug and there's no cure. It's a very serious and disabling illness. An estimated 2/3 of individuals indicate onset followed a flu-like illness. There's what appears to be a hidden pandemic of ME/CFS to have occurred in the mid-'80s, including multiple outbreaks in the US. The outbreaks have been traced back to the 1910s and suspected even earlier. Many of these are associated with polio outbreaks. A lot of that information is in the books by Byron Hyde. But I'd like to tell you a little bit about the epidemic that occurred in the US in the mid-'80s. Most of this information that I'm going to tell you comes from the definitive history of the mid-'80s outbreaks in the book Osler's Web, which is written by investigative journalist Hillary Johnson. In 1989, about 1,000 calls a month about chronic fatigue syndrome were being received at the CDC. This actually outnumbered the calls about AIDS. It was known to people that the CDC knew something about chronic fatigue syndrome because in 1988 they had actually named this mysterious syndrome chronic fatigue syndrome, which is a trivializing name that the patients don't like, and that's why we're using ME/CFS. By 1990, the CDC was receiving over 7,000 calls a month and 1,000 letters a day about chronic fatigue syndrome. And the CDC had to hire a full-time employee to record the names and addresses so they could be sent a bulletin telling them there was no convincing evidence that that chronic fatigue syndrome was contagious, and that only a small number of patients have been found to have modest immune system differences. So unfortunately, there was no investigation of these outbreaks, so we don't know what virus or viruses caused them, although it seems very likely that the same virus or its variants caused all those outbreaks in the mid-'80s. Now, the prognosis for ME/CFS is very poor. Only about 5% recover to normal activity levels. And the most severely 5% are completely bedbound, and some are tube-fed, not able to speak, nor can they tolerate sound or light. And one of those victims is shown there on the bottom. So how do you diagnose myalgic encephalomyelitis chronic fatigue syndrome? A report from the Institute of Medicine came out in 2015 that is recommended 4 of 5 symptoms to diagnose ME/CFS. This is cognitive impairment, orthostatic intolerance, post-exertional malaise, unrefreshing sleep, and new fatigue of greater than 6 months. And if you have 4 of those symptoms, then you can be diagnosed as ME/CFS. And as a result of that, some people are saying that long COVID individuals have ME/CFS, but we have to remember that in 2015, we didn't know about long COVID, and so it's hard to know what diagnostic criteria would be used if it had been done in light of knowledge of long COVID. There are additional symptoms that are shared by ME/CFS and long COVID. Many of these are familiar to you with joint pain, muscle pain, GI distress, sore throat. And there are, however, some symptoms of long COVID that are fairly rare in ME/CFS. One is chest pain and pressure, a cough, reduced sense of smell, rash. So there are some differences, and this needs to be explored further. So I just want to give you a brief history of the ME/CFS outbreaks. There have been 75 documented outbreaks, but a huge number of those outbreaks that caused those 7,000 people to call into CDC have not been documented. The problem was in the mid-'80s there was no social media, so when somebody called in, they didn't know about how many thousands of other people were experiencing the same thing. But there were some documented outbreaks, especially if it happened in a small location, it became evident. So in the 30s and 40s, there were outbreaks documented, and wherever you see red there. But in the 50s and 60s, there were a lot of these outbreaks, and in different places they were named different things, and that's scrolling through a number of these large numbers of names that were used. But in the 50s, there was an outbreak at the Royal Free Hospital in London, where a physician there named the illness myalgic encephalomyelitis for the muscle pain and what was thought to be inflammation of the brain and spinal cord. The main reason this has not been accepted is that there's still a question as to whether there is inflammation of the brain and spinal cord. For ME/CFS from long COVID. And one of them is that symptoms of long COVID can arise after an asymptomatic or mild case of COVID-19. And also that long COVID may appear weeks after the acute infection seems to have ended. So another lesson is that the diversity in the post-viral symptoms of different individuals that we're seeing in ME/CFS doesn't mean that there are multiple different genera of viruses inciting a post-viral illness. I think many of you here who may work on long COVID know that there was a recent paper showing that there seemed to be 4 clusters of symptoms in long COVID. And so we're seeing this in ME/CFS, and people are saying, oh, that means there's different viruses caused it. That doesn't mean that. We don't know. There are some lessons for long COVID from ME/CFS as well. And that is that the viruses that caused ME/CFS prior to 2020 are still causing the same illness after 2020. And so you can't assume that individuals who fulfill ME/CFS diagnostic criteria actually have long COVID. Long COVID is actually more prevalent, but you're going to have some people also who actually have pre-2020 ME/CFS. And we really need to compare ME/CFS and long COVID at the molecular level if we want to determine whether or not it was actually SARS-CoV-2 that was the inciting virus when you have this person who fulfills the IOM committee criteria for ME/CFS. I would like to say that pre-2020 ME/CFS is a post-viral illness. A majority of ME patients identify an infection as preceding their long-term condition, immediately preceding. Now, the hypothesis that many different families can cause ME/CFS is often stated as fact, but actually we don't have evidence for that. We don't know whether there's more than one virus family that causes it or not, and that's reviewed in a recent article in PLOS Pathogens that I wrote. So what about those who suspect non-infectious causes? A number of people, if you ask them, how did you get ME/CFS, they'll say, oh, I was under terrible stress and then I got ME/CFS. Well, people look for reasons for why they become ill, but the fact is that most ME/CFS without obvious preceding illness could have arisen from asymptomatic viral infections, just like they're arising in long COVID, and not from emotional stress or chemical exposures. We don't really know. So people with ME/CFS have symptoms that act as if they have a viral infection. You know, these are symptoms you might have if you have a viral infection. So I will like to mention that the enterovirus family is the one that's most often implicated in ME/CFS outbreaks. And at the bottom there that you can't see, there's an article written by a student in my lab and I reviewing in Frontiers of Medicine the evidence for enteroviruses. There's also known that there's reactivation of herpes viruses that are observed in these outbreaks of ME/CFS, and of course that's been seen in long COVID as well. So I'd like to point out that both enteroviruses and SARS-CoV-2 can cause long-term infections. A lot of people don't realize, and in fact didn't realize before SARS-CoV-2 came on the scene, that you can get a long-term infection from an RNA virus, and I think maybe we'll be hearing more about that at this meeting. But you can find enteroviruses in humans as well as in cell lines. So what are the leading theories of the underlying cause of ME/CFS? One of them is structural damage that affects neurological or other functions, pathogenic autoantibodies, a persistence of some sort abnormal biochemical pathways or gene expression, maybe some sort of epigenetic change, and also a chronic viral infection, which could either be the inciting virus or reactivated viruses that result from this infection. And those of you who work on long COVID will realize this is the same list that you probably see when you're at a long COVID meeting. So is there any evidence for chronic viral stimulation of the immune system in the pre-2020 ME? And it could, I'm going to be agnostic as to what this virus could be. It could be an inciting enterovirus or some other virus. It could be reactivated herpes viruses or even activated endogenous retroviruses, just asking, is there any evidence for chronic viral stimulation? So I'm going to tell you three things, work from my lab and work in our center about metabolism of immune cells, markers for immune cell exhaustion, and activation of classical monocytes. So as most of you know, T cell metabolism changes following stimulation of the T cell when it detects a virus or some other pathogen. And the quiescent T cell has a lot of fatty acid oxidation, and your proliferating T cell, you upregulate glycolysis and oxidative phosphorylation in healthy individuals. But in ME/CFS, the glycolysis goes down, is lower in ME/CFS when you assay CD8 T cells. Mitochondrial membrane potential is lower in cells from ME/CFS patients. And fatty acid oxidation is increased. And these are all hallmarks of potential T cell exhaustion, certainly T cell abnormalities. So long-term stimulation by chronic viral infections is known to cause T cell exhaustion. And then the question is, are CD8 T cells in an exhausted state in ME/CFS patients? So you can identify exhausted cells by the presence of inhibitory receptors and specific transcription factors. And the ones that we've analyzed in this initial study was PD-1 and also the transcription factors TCF1 tox and TBAT. And the protein tox is an example of a transcription factor that drives exhaustion. And so if your transcription factor is active, it can increase the expression of immune suppression markers like PD-1, and it can also decrease the expression of other genes so that the immune response is reduced. So in this study, which was done by my graduate student, Jessica Maya, we started with 15 ME/CFS patients and 14 healthy controls. She isolated CD8 T cells and then examined the prevalence of these proteins using fluorescent antibodies. Now, I'm only, given time, I'm only going to show some of this data. This is unpublished, but we are writing it up now. So this just shows T cell development. We did look at different stages. I'm going to start with the total CD8 T cells. And if we look at the prevalence of cells that have both PD-1 and TCF1, you can see that it's higher in the ME patients. Cells and CD8 T cells. Also, there's a higher proportion of cells that have PD-1 tox and TBAT, and there is a higher proportion of these cells with all four of these proteins. Then if we look at the assaying early differentiated CD8 T cells using markers to identify which ones are the early differentiated ones, again, we see higher tox in these early CD8 T cells, and we see a higher cell proportion that has PD-1 tox and TBAT. And finally, if we look also at terminal CD8 T cells for the frequency of tox, again, you can see that the ME/CFS cells have a higher proportion of tox compared to the controls. And this is interesting given that T cell exhaustion is being considered in long COVID as a mechanism, and as shown in this diagram, it potentially could either be SARS-CoV-2 still replicating, or some antigen RNA still there that is activating the immune response, or again, just like in ME/CFS, there could be a latent virus reactivation that is causing this exhausted immune response. Of course, a combination may be what's happening as well. So what other indications can be obtained from immune cell analysis? I now like to talk about some work done by my colleague Andrew Grimson, who is the associate director of our center. His lab did single-cell RNA sequencing of 30 ME/CFS patients and 30 sedentary control subjects with 4,500 cells RNA sequenced for each subject. This work is available as a preprint at bioRxiv. So this UMAP here shows an analysis of the genes expressed in individual cells and then clustered so that you can, and then you can identify which of these clusters corresponds to which cell type. And when he looked at this, the most dysregulated cell type was classical monocytes. So we'll look a little bit more at classical monocytes if you compare ME/CFS and controls. And just to give you the bottom line before I show you some data, the classical monocytes are expressing genes indicating they're being signaled to develop into macrophages, which then go to tissues and are responsible for killing pathogens or infected cells. The pathways dysregulated in classical monocytes are listed here, and all of these pathways, the 5 that I've listed there, this is consistent with what I just said, increased monocyte migration to tissue. So this is from the single-cell RNA-seq. This was validated by purifying classical monocytes from four cases and 4 controls, and then comparing the transcripts by RNA-seq. And the transcripts that were the most dysregulated were these CCL4 chemokines, which in fact entract immune cells to go through the endothelium and migrate into peripheral tissues. So another very interesting analysis that his group did, and I think fairly unique, was a machine learning analysis to classify each cell from its RNA sequence, whether it was, quote, normal or diseased. So here's an example of a UMAP in which we're showing one case and one control, and all the dots that are blue are the cells that are predicted to be normal, and the ones that are yellow or orange are the orange are predicted to be normal, and the blue is predicted to be diseased. So if you look at quite a number, if you look at all these ME/CFS cases, you can see that there's some variation in the monocyte dysregulation. There's some people with very blue configuration. There's some that are more orange, but all the controls look quite orange. And in fact, if you compare the percent of classical monocytes that you predict to be disease, there's really quite a separation between the cases and the controls. And even more interesting, the number of these predicted diseased classical monocytes correlates with the extent of fatigue according to a fatigue scale filled out by the patients. So to summarize, I've told you that outbreaks and patient reports indicate that pre-2020 ME/CFS is a post-viral illness, and that there are immune abnormalities that distinguish ME/CFS and control subjects, and that these immunometabolism abnormalities is consistent with T-cell exhaustion. Expression of PD-1 and transcription factors are even more evidence that T-cell exhaustion is occurring. And classical monocytes in ME/CFS appear to have received signals that will induce them to become macrophages to battle against an unknown antagonist that, unfortunately, unlike SARS-CoV-2, we don't know what it is. I'd just like to end with my acknowledgments. The people in my lab who did this work, Jessica Maya and Alex Mandarano, the subject volunteers and our physicians who diagnosed the cases of ME/CFS, as well as our donors. And Andy Grimson's group is shown there as well. Thank you very much.